Alright, so biomolecules, these are living molecules, and biomolecules are supposed to understand water, you're supposed to understand proteins, carbohydrates, lipids, supposed to understand also enzymes and nucleic acids. You've done carbohydrates, we are doing proteins. Next time we'll do lipids and enzymes and then nucleic acids. So we want to describe what a protein is. So when you're talking about a protein is an organic compound. If you hear of organic compounds, these are compounds made up of carbon and hydrogen. Those are organic compounds. So a protein is an organic compound. It's going to be made up of carbon, hydrogen, oxygen, and nitrogen. It can as well include sulfur sometimes. Sometimes they can just ask you the atoms that make up a protein, so make sure that you know them. Okay. Now, proteins are going to be made up of small molecules and we call them monomers. Okay, a monomer. So what is a monomer? A monomer, these are simply molecules that you have to put together for it to form a bigger molecule, and that bigger molecule we call it a polymer. And some molecules that have got monomers, carbohydrates have got monomers, and the monomers of carbohydrates we call them monosaccharides. Proteins also have got monomers. The monomers of proteins are called amino acids. Enzymes also have monomers. That is because enzymes are also made up of proteins. Also nucleic acids. So of the biomolecules, it's only lipids which don't have monomers. Like you not say if you get this molecule, you put it together, you put it together, you put it together, then you form this lipid. No. You make lipids from different molecules. For monomers, you get the same molecule, you put a lot together, and then you form a bigger molecule. Does that make sense? Okay. Now let's first understand the functions of proteins. I mean, so first function of proteins. Proteins can be used in structure provision in structures. And an example of such a protein that is used in structures is collagen. So collagen is found actually in the skin, so it is used in structures. Proteins are also used for transport, and an example of a transport protein that we can talk about is hemoglobin and myoglobin. These they get to take blood in blood. So hemoglobin transports oxygen in blood, myoglobin transports oxygen in muscles. Another function is that the proteins can be used as enzymes, so enzymatic activity. Enzymatic activity. An example that we can mention things like maltase, sucrase, those enzymes. And then the same proteins can be used as hormones. For example, the hormone insulin. Insulin is a protein and it's an hormone. Hormones are basically molecules that regulate or control activities. For example, if it is time to menstruate, you need an hormone to cause that to happen. If it is time to start developing characteristics to look as a mature girl, you need hormones. Hormones are also necessary for protection. Example of such hormones used for protection are antibodies. Antibodies are hormones used for protection. They're gonna just ask you the hormone used for this is, they can mention anything there. So make sure that you know that. Now remember we are saying that hormones are made up of monomers, and the monomers for proteins we call them amino acids. So proteins will come from amino acids. That is the basic building block of proteins. You need to know the basic structure of an amino acid. An amino acid is going to have a carbon. This carbon is bonded to another carbon. This carbon which is bonded double bond to oxygen and then also to OH. And then this carbon is also bonded to a nitrogen, which is bonded to two hydrogens, like this. This same carbon here is also bonded to a different group, R group, and then it's bonded also to hydrogen. So you can see that an amino acid is bonded to four different things. What are the groups? This group that you can see here, we call it an amino group. This group that you can see here is the carboxylic acid group or the carboxyl group. That's why we call them amino acids, because they've got a carboxylic acid and then an amino group. So carboxyl or carboxylic acid group. And they are also having an hydrogen, and then they also have a group here which we call as an alkyl group. Okay, it's basically a chain of carbons bonded to hydrogens like that. Now this alkyl group can differ, and it is going to be the one that will determine the function of that amino acid. Very, very important. Okay, the differences in the R group determines what type of amino acid that you have. Another way in which we can write an amino acid is we can actually write carbon. On top we can still write R, down we can still write H, but this side we write C. You can see this C is bonded to O and then OH, so you can write it as CO and then OH. And then this side we can have N and then we write two hydrogens. So we can as well write it like this is the same an amino acid. There are 20 amino acids available, and all these 20 amino acids, the R group is going to be different. The simplest amino acid is known as glycine. This one you need to know it by heart. Simplest amino acid is glycine. And glycine, the special thing is that the R group for glycine is an hydrogen. This one you need to know. Is this okay? Okay, good. Now, because of this, because remember we are saying that this carbon on the middle is supposed to be bonded to four different things. So for glycine, it's not going to be bonded to four different things. When you have got a carbon bonded to four different things, to A, to X, to B, and to Y, four different things, we call this carbon as a chirocarbon. It is also known as asymmetric carbon. But if a carbon is bonded to four things, but then they are not different, maybe you have got two which are the same, that carbon is known as achiral, also known as symmetric. Okay, very important. So because glycine is the simplest amino acid, compounds which are chiral or symmetric, they have got optical activity. So we call them optically active. What do we mean if we say optically active? They contain light. So if you get an amino acid that has got a carbon bonded to four different things, you put it in solution and then you want to pass light there. Okay, let's say you put it in a bottle and then you want to pass light through. Light is going to turn. That's the meaning of optical. Optical means light. So it is optically active, means it can turn light. But glycine, because it is achiral or symmetric, it is optically inactive. It can't turn plain polarized light. Light will just pass through it. That is very important for you to take note. Very, very important. Okay. What else do you need to know? So this carbon that you can see here, we call it as a alpha carbon, this one. Okay. That's known as the alpha carbon. And then this other carbon bonded to the alpha carbon is known as a beta carbon. Now you need to know that most of the proteins or most of the amino acids are found in the L form, because you can find amino acids is in the D form or the L form. Carbohydrates are mostly found in the D form. It's opposite to proteins or amino acids. They are mostly found in the L form. Take note of that exam question. All right, that is very important. So we've already mentioned the simplest amino acid, which is symmetric, okay, and it is optically inactive. So we've talked about that. And we are saying that we have got 20 amino acids. How do these 20 amino acids differ? They differ by the ara group, just the ara group. Now, of the 20 amino acids, you are going to have amino acids which we call essential. Other amino acids are non-essential. Any idea what do we mean if we say essential amino acids? Any idea? Okay. So essential amino acids, these are amino acids which you can't make. They have to be gotten from the food that you eat. Same with essential fats. Every time you hear essential, you can't make it. Non-essential, it means that your body can make it, and so you don't need it from food. Is that okay? Okay. Now you can classify amino acids in different ways. Let's talk about the classification, and this is very important. You can classify amino acids in different ways. Some amino acids can be attracted to water, or they love water. What is the term that we use for things that love water? Yeah, hydrophilic. Very good. So some amino acids are actually going to be hydrophilic, meaning they love water. Now, the only way something can love water, can be hydrophilic, is it should be polar. Anything that is non-polar is hydrophobic, because things which are non-polar, they can't dissolve in water. So one classification of amino acids, we can talk about them if they are polar or non-polar. Do they love water or they hate water? Another classification of amino acids we can use, we can classify them if they are straight or if they are branched. So some amino acids are straight and we call them aliphatic, while other amino acids are in a circle and we call them aromatic. Okay, so that is another classification. We can as well classify some amino acids if they are acidic or basic. So we have got acidic amino acids and we have got basic amino acids. And we can as well, some amino acids contain sulfur, so we can say sulfur containing. While some other amino acids are actually going to be, they will not fall in any one of these classifications, so we call them amino acids, and we only have one amino acid. So take note of these classifications, but what I want you to do is you don't need to master this. You need to know why we classify an amino acid like that, because there's no need for you to swat or anything. When you look at a bond between a carbon and hydrogen, is this bond polar or it is non-polar? From what you think? Okay, it's actually going to be the opposite. So every time carbon is bonded to hydrogen, those bonds are non-polar. So if you see a lot of carbon to hydrogen bonds, just know that this molecule can't dissolve in water. And because it can't dissolve in water, it means that it is non-polar, it is hydrophobic. That is why, lipids have got a lot of carbon hydrogen bonds. So lipids don't dissolve in water. They're hydrophobic. The reason is they have got a lot of carbon to hydrogen bonds. But if you see a bond between oxygen, fluorine, nitrogen, any one of these with hydrogen, these bonds are polar. Okay, and so you expect that amino acids which contain them to be also polar amino acids. That is very important. Very, very important. And then you are going to have some other amino acids which are going to be acidic. But then, how do you talk about acidity? When you see an amino acid where the R group, so all these things I'm talking about is depending on the R group. If you see the R group has got like this group, do you think it's going to be acidic or basic? What do you think? Very good. That is because this is carboxylic acid group. How about if you see an amino acid where the R group has got a lot of amino group? Is this amino acid going to be acidic or basic? Basic. Just that. That is a property that you need to have in mind because they don't require you to master anything. Let's write the basic structure of an amino acid. You have got a carbon bonded to COOH and then bonded to NH2, then bonded to hydrogen. Remember, it's supposed to be bonded to an R group. But what I'm going to be doing is I'm going to be changing the R group and then we'll comment whether it's going to be acidic, basic, or anything else. The simplest amino acid, remember we said that is going to be, what is the name? Do you remember? The simplest amino acid? Glycine. Glycine. You remember? So glycine has got an hydrogen here. Do you think this amino acid is going to be polar or non-polar? So because it has, it's non-polar, very good. So glycine is a non-polar amino acid. And you can see it's not in a chain. Is it going to be aliphatic or aromatic? Aliphatic means straight. Aromatic means it has got a ring. But it doesn't have a ring. So unless if it has something like this, it's aliphatic. Good. Yes. So let's talk about another amino acid which is known as alanine. Now alanine has got a methyl. It has got CH3 here. Do you think it's going to be polar or non-polar? Non-polar, very good. How about, is it aliphatic or aromatic? It's also aliphatic. There's no ring. Very good. How, let's talk about valine. Now, valine, the group is known as isopropyl, so it has got a group like this. It has got a carbon bonded to CH3, bonded to CH3. Do you think this amino acid is basic or, I mean, polar or non-polar? It's non-polar. It has got a lot of carbon hydrogen bonds. Is it aliphatic or non or aromatic? It is still aliphatic. There is no ring. As long as you can't see a ring, it can be aromatic. Is that okay? Okay, good. Let's talk about leucine. So these are the 20 amino acids we are talking about. So leucine has got a isobutyl group. So it has got like this CH2, CH, CH3, CH3. Do you think this is polar or non-polar? Hmm. What do you think? So it's going to be non-polar because it has got a lot of carbon hydrogen bonds. That is going to be the same also with isoleucine is also non-polar. Let's talk about serine. Now serine, because serine contains an OH, can as well classify them among those amino acids that have got an alcohol. So we call them hydroxyl containing. Just like we have got sulfur containing, we also have hydroxyl containing. Do you get that? So this is known as serine. Serine contains an hydroxide. So now let's see. Is this amino acid going to be polar or non-polar? It's polar. Why have you said it's polar? Okay. Uh-huh. That's a reason. Bonded to oxygen. A bond between oxygen and hydrogen is always polar. That's the reason. So apart from serine being the one which contains OH, you have got another amino acid that contains OH, and that is known as the threonine. So for threonine, what you're going to see is that you have got a carbon bonded to an hydrogen, it's bonded to OH, and then bonded to CH3. It's also hydroxyl containing. Do you think it's polar or non-polar? It's also polar. Excellent. Okay. And they are all aliphatic because there's no ring there. Yes. Let's talk about asparagine. Asparagine, you are going to have CH2 bonded to CO and then bonded to NH2. Do you think it is polar or non-polar, asparagine? It's polar, excellent, because you can see bonds between nitrogen and hydrogen, right? And carbon and oxygen, those are polar bonds. Okay, good. So this is known as asparagine. It is polar and it is aliphatic. Let's write another one, pending with the chain that we have on top there. Say we talk about, okay, glutamine is also going to be polar because it's also going to have CO, okay, like that. Let's talk about this one, phenylalanine. Phenylalanine. So phenylalanine is going to have a CH2 here, and then bonded to it, you are going to see a ring like that, and then it has going to have OH on top. Is this one polar or non-polar? It is polar. Why is it polar? Very good. Is it aliphatic or aromatic? Very good. It's aromatic. Okay. So that is how you're going to be able to classify it. And then let's look at one more. Okay, we can look at some other few examples. So I've got CH2, and this one is bonded, let's say, to a ring like this one. Let's say it looks something like this. Okay, so something like this. So this amino acid that I've written here is known as tryptophan. When you start looking at genetics, it's actually one of those few amino acids which are only going to have one codon, tryptophan. Okay, so commenting on the polarity, do you think tryptophan is polar or non-polar? What do you think? Okay, let's comment on, is it aliphatic or aromatic? What do you think when you comment on that property? So it's going to be aromatic. Okay, so that is for tryptophan. So you can look at other compounds, and so we've just been talking about aromatic, aliphatic, and also polarity. But when you see something that will contain a sulfur on top, that is going to be sulfur containing. And the only two that you need to know which are sulfur containing, there is methionine and cysteine. These two, you just have to know them. And you also have one which you call an amino acid. It's only one, it's proline. Acidic and basic amino acids will depend with the kind of group that you have. So those which have got OOH, they are acidic. For example, acid. Aspartic acid is like this, CH2 and then COOH. This is aspartic acid. So this one is an acidic amino acid. Same with glutamic acid. Now, aspartic acid is also known as aspartate. Don't forget that we also have an amino acid known as asparagine. This one is aspartate. Okay, not asparagine. And then glutamic acid is also known as glutamate. Don't forget we also have what we call glutamine. It's not the same. This is glutamate. Okay, so glutamic acid is uncharged. When it becomes charged, it's known as glutamate. Same with aspartic acid is uncharged. When it becomes charged, it's known as aspartate. So these two that I've mentioned, these are acidic amino acids. We also have basic amino acids, and basic amino acids will include histidine, arginine, lysine. So when you look at their structure, you are going to see nitrogen in their structure, histidine, and those others that you've mentioned, histidine, lysine, arginine. Let's just see one, see how the structure looks like. So if we look at how arginine is, we are going to have CH2, there are actually three of them, and then you have got NH, and then it's bonded to a carbon, bonded to NH2, and then double bond to NH. You can see you have got a lot of nitrogens. If you just see a lot of nitrogens, the structure, just know that that one is basic. So this is how you get to classify amino acids. That is very important. Nothing that you need to know is you can connect a lot of amino acids together, and that is going to form a peptide bond. And the peptide bond is what ends up forming a protein. How do you form a peptide bond? You have got a carbon bonded to R, to hydrogen, to nitrogen bonded to two hydrogens like this. Let's have a carbon here bonded to oxygen like that. Remember, it's also bonded to OH. I'm going to write another amino acid. This side, let me write it like this. I write hydrogen down, then hydrogen this side. Can have another carbon bonded to R, bonded to H, and then carbon, double bond to oxygen, and then OH, and then we have got nitrogen bonded to hydrogen and then bonded to hydrogen. So these are two different amino acids. To form a bond, what will happen is that these two are going to come together. OH from the carboxyl group of one amino acid will join with hydrogen from the amino group of another amino acid. And in the process, you are going to release water. So this is going to go. So you are going to form water. And then this bond is going to be formed here. Remember, this reaction produces water. So this reaction is known as a condensation reaction. The other term is for condensation reaction is that it's an hydrolysis reaction because we produce water. Okay, so that is how we get to form amino acids. So you only have two amino acids here. This bond that you see between amino acids is known as a peptide bond. What are the atoms that we found on the peptide bond? It's carbon, oxygen, nitrogen, and hydrogen. When we have got two peptides, we call that as a dipeptide, three tripeptide, four tetrapeptide. But if you have got two to 20, we call that as an oligopeptide. And when you have got more than 20 amino acids, greater than 20, we call that as a polypeptide. That is how we get to form these, right? And so when you get to put these polypeptides and they become functional, that will end up forming now a protein. So that is how we get to form these ones. Now we have got what we call the levels of protein structure. But before I talk about the levels of protein structure, there is something that we need to know, which is known as the Zitterion nature. It's a property of an amino acid. What amino acids do is that, remember the structure of an amino acid: a carbon, R group, hydrogen, a carbon, double bonded to oxygen, OH, and then you have got nitrogen bonded to hydrogen, and then to hydrogen. I've written it so big. So this normally in neutral form, what happens that this hydrogen is not attached to this end. So this hydrogen actually goes and attaches to the amine group of the other amino acid. Now let me write it properly. So what you are going to see, I'm going to write three forms. I've got carbon bonded to R group, to hydrogen, carbon, double bonded to oxygen. It's only going to have O, okay, like this. You can see how I've written it. And then here, now you have got three hydrogens. Where is the third hydrogen coming from? It's the one which was on here. So this is going to be positively charged. So amino acid existing both positively charged and negatively charged is known as a zwitterion. So this is known as the zwitterion nature of an amino acid. Now guess what happens when you add acid to an amino acid and when you add base to an amino acid. So this side, let's say we are adding base. This side we are adding acid. How does it become? When you are adding base, the characteristic of a base is OH. What will happen is that that OH is going to react with one of these hydrogens. Okay? So meaning that you are not going to have three hydrogens. So what you end up with is a carbon bonded. Let me write it like this: COO with a minus. I've written this side. On top you have R, down you have got H. This side you're only going to have any H2, and there's not going to be any positive charge. So in an alkaline environment, amino acids are negatively charged. But when you add an acid, what does acid mean? We are adding a proton. Okay, here we are adding hydroxide. When you add a proton, this proton is going to be attracted to the negative O. And so what you are going to have is a carbon bonded to R, bonded to H. And then you are going to have COOH. But this side you still have N with 3 hydrogens is positively charged. So in an acid, amino acids are positively charged. So it's important for you to take note of the charge of the amino acid in what environment. Amino acids have got levels of protein structure. You are going to have the primary level, the secondary level, the tertiary, and the quaternary. You need to understand what they are and some examples is very important. Okay, so what are we talking about? The levels of protein structure. So on the levels of protein structure, the first level is known as a primary level. What really is a primary level? Primary level just describe the amino acids that is forming a protein. What are we talking about? Just arrangement, that's all. And arrangement of what? Arrangement of amino acids which are forming a protein. For example, you have got amino acid 1 bonded to amino acid 2, bonded to amino acid 3, bonded to amino acid 4. So the only bond that you find in the primary level is known as a peptide bond. What is a peptide bond? It is a bond that is found between two amino acids. That is the only one. And then if we go to the next level of protein structure is known as a secondary level. So what really is there in the secondary level of protein structure? Now in the secondary level, the amino acids that have formed a peptide, a chain, will start to coil about themselves. So they can coil into two, they can form what we call an alpha helix or a beta pleated shape. These are the two structures that they can form. And how do these look? An alpha helix is like a coil. So you'll be able to see amino acids like coiled like that. So that is alpha helix. And a beta pleated shape is something that looks like this. Okay, so these are the two shapes. One thing that you need to know about the secondary structure is that it is only made up of one peptide or polypeptide. It's only one polypeptide. For example, you add to join 150 amino acids and then now they start coiling about themselves. They start coiling. So just like that. It's very important. So you need to know at least an example of a protein that is in each form. Alpha helix, example that we can talk about is keratin. Keratin is alpha helix. Okay, it's an important one. So this keratin is found in hair, it's found in wood, in nails, in the beaks of things like birds. That's where you find the alpha helix. And then you also have the beta pleated shape. The question should be, where can we find the beta pleated shape? This one is going to be found in things like silk, fibroid. Okay, silk. This is going to be produced by silkworms and yeah, produced by silkworms. That's where we can find these. Now there are different bonds that will be able to stabilize the secondary structure. So apart from peptide bond, you can't have secondary structure without primary. So meaning that secondary structure also has the peptide bonds. But apart from peptide bonds, we have got additional bonds, and those include things like hydrogen bonds. And actually, hydrogen bonds are the most important, okay, when you're talking about a secondary structure. The third structure is known as the tertiary structure. Now, what is a tertiary structure? In a tertiary structure, what you're going to see is that there's going to be bonding and bending and folding of proteins, and that will be able to form again different structures. You can either form a fibrous structure or you can form a globular structure. Okay, so something that is fibrous, for example, collagen is fibrous. Or something that is globular, like you are going to have some proteins which are like that, coiled around themselves, they look like a ball. For example, enzymes work well when they are globular. If they just lose their globular shape, they will no longer be functional. Okay, that's the meaning of globular. Now, there are a lot of bonds that hold again the tertiary structure. Because tertiary structure is made up of secondary structures, it should have peptide bonds, it should have hydrogen bonds. Besides this, in tertiary structures, we also have what we call disulfide bonds. These are bonds between sulfur atoms, like for example cysteine. We also have hydrophobic interactions, because you remember we have got some hydrophobic amino acids. Apart from that, we also have van der Waals forces, okay, van der Waals. We also have some form of ionic bonds, okay. So all these bonds are going to be found in the secondary structure. And the last level of protein structure is known as the quaternary level. Now, the quaternary level is basically an aggregation, or you are bringing together different polypeptides which are held together by all these bonds that you can see here. For example, you have got this one polypeptide, and then another polypeptide, and then another polypeptide, and then another polypeptide. You bring them all together, okay? And then that forms one protein that is known as a quaternary structure. And examples of quaternary structures we can mention of hemoglobin. Hemoglobin is made up of four chains. Okay, it has got four chains, of which two are alpha chains and two are beta chains. They have got amino acids. The alpha chain has got 141 amino acids, the beta has got 146 amino acids, but they are two, and when you put them together, you form this which we are talking about to be hemoglobin, which is a protein that carries oxygen. You also have insulin. Insulin is an important protein that regulates glucose. Okay, so when there is too much glucose, for example, if eaten food that contains sugar, insulin will make that sugar to be stored as glycogen in the liver or in the muscle. And when glucose becomes low, you are going to have insulin promoting the breakdown of glycogen. All right, so that is what is going to be able to happen. You also have actually insulin will work in control with what we call glucagon. So when you don't have enough glucose, glucagon is going to cause a breakdown of that glycogen. Okay, so insulin is converted into dimers, so it has got two peptides, and then into hexamers, into six polypeptides, and these are going to be held together around zinc. So like you have got a zinc on the middle, and then you have got now these six polypeptides like that. Okay, And insulin is secreted in the pancreas by what we call the pancreatic beta cells. You also have collagen. Now, collagen is a fibrous protein. I had mentioned it here. And this protein forms a triple helix. It has got like a three, okay, like a triple helix. It's here. So that's why some questions you see they'll ask you to say what is the most important structure for collagen. So that is like secondary structure. It is a triple helix. The secondary structure is the most important, and it is going to form fibrils, long fibrils, which form more fibrous tissue. So collagen is actually found in strong places like in bones, tendons, these things that you can't break easily. So collagen is made up of three alpha helix chains. You are going to have one like this, and then another one is going to pass like that. Then you are going to have the third one, three of them. So that's collagen. Remember that the most important structure for collagen is secondary. So this is it on the amino acids. So let's just answer some questions. The first question here is saying essential amino acids are the 20 naturally occurring amino acids are found only in animal tissue, can be synthesized by the human body, cannot be synthesized by the human body, are found in plants and not in animals. Which one would we pick? Okay, so when you're talking about essential amino acids, these are amino acids that cannot be made by the human body. You have to get them from food. So they cannot be synthesized in the human body. They can be found in other animals, not just in humans. All right. Choose the term that explains the ability of amino acids to function either as acids or bases. Now, functioning as both acids and bases, that is amphoteric. Do you know the meaning of antiphatic? It's being both hydrophilic and hydrophobic, isn't it? In an acidic medium, amino acid glycine, which is given here, so they've given us amino acid glycine looks like this: NH2, CH2, COOH will exist as. How will glycine look in an acidic medium? Remember we said that in an acidic medium, what are you donating? Acids donate hydrogens. So those hydrogens are going to go to the amine, which is what? Positively charged. So end up having three here, and then nitrogen is going to be positively charged. So this is how it's going to exist. So which one is that? So that is this option here. Okay. The hydrogen is still there at the end. Choose the statement that is true about amino acids. Amino acids produced by human cells are called essential. This is not true. Amino acids with two amino groups are called basic. That is not true. They are known as what? With amino acid with two amino groups. Okay, this can be true with two amino groups. One amino group, remember, one amino group is this one here. So we are guessing that the other amino group is actually here on the other group. And then they're going to be basic. So I'm going to still come back to this option. It can be true. Amino acids with two carboxyl groups are called acidic amino acids. So meaning that if, okay, this is also supposed to be true, because if you have got carboxyl group here and also a carboxyl group here, that's true. The most common form of amino acid is the D. No, remember we said it's L. So the answer is 2 and 3 above. The primary structure of protein is maintained by, so we had mentioned to say only, not the primary structures, not hydrogen bonds, not ionic, not phosphodiester, not disulfide. It's actually covalent bonds, and these covalent bonds we call them peptide bonds. Okay, peptide bonds are a special type of covalent bonds. So the following chemical group is not a component of all amino acids. Amino group is a component, carboxylic group is a component, R group is a component, hydrogen is, but not an ester group. So that's the answer. Secondary structure of a protein is determined by, remember this is secondary, number and order of amino acids. The number and order is primary. Hydrogen bonds made between CO of one polypeptide and NH of another. This is true. Okay, you're going to have hydrogen bonds. Disulfide bonds, this is going to be in tertiary. Non-covalent bonds, that's tertiary. These are the hydrophobic bonds. Association between different polypeptide chains. Actually, secondary, I had mentioned to say it should be the same polypeptide. I did mention that, so this is not true. So the following compound is not a polymer. Just at the beginning, we had mentioned this. Lipids are never polymers. What is the role of the SH group of cysteine in the structure of protein? Forming disulfide bonds between pairs of cysteine in different parts of a polypeptide chain. Is going to be true. Forming disulfide bonds between cysteine and different amino acids, really not. Disulfide bonds are supposed to be between atoms that contain the sulfur. Other amino acids might not contain, because remember we only have methionine and cysteine as the ones to contain sulfur. Forming disulfide bonds through reduction, really not. Forming disulfide bonds through oxidation of the SH. Okay, do we oxidize it? Okay, I don't know about oxidation. Maybe it is, you can get to check it, but disulfide bonds is not oxidation, it's just that attraction. Peptide bonds which covalently link two acids results from oxidation, reduction, hydrolysis of, is supposed to be hydrolysis of amino acid, condensation of amino acids, formation of hydrogen bonds. So what are we saying? Peptide bonds which covalently link two acids. Okay, it results from condensation, right? Yes. Condensation will remove water. So I'd mentioned the word hydrolysis. That was wrong. It is known as condensation or dehydration. Hydrolysis is when you are breaking down proteins, when you are reacting them with water. So that is supposed to be dehydration or condensation. Which of the following levels of protein structure describe three-dimension of folding of polypeptide? So three-dimension of folding is quaternary. Secondary is 2D. Primary is just amino acid. Tertiary is just putting amino acids together. Okay. Actually, tertiary structure also represent, because we are putting quaternary structures together, so it also represents the 3D. So the answer is both 1 and 4. Which of the following amino acids has or have a hydrophobic side chain? So let's be looking at the side chain. The side chain is this one here. Is that hydrophobic? No. This other one down here, is it hydrophobic? Yes, you can see you only have carbon bonds. How about this one? Is it hydrophobic? Yes. How about this other one? Is it hydrophobic? No. So it's supposed to be B and C. So that is the answer 3. Alright, so that is it for this one. Thank you so much.